In this video, we will learn about gene expression and DNA microarray analysis. In biological sciences, we mainly deal with living organisms, and the key to understanding a living organism is to understand the expression of its genome. Further, we should also know what types of genes are present and what types of proteins are produced when the genes are expressed. Beyond this, we may want to understand what is going on in a particular cell within an organism. How does a particular cell type perform gene expression? And besides this, we may also want to know how has gene expression been altered in any diseased or stressed cell. A common approach for answering all these questions involves identifying mRNAs being made by a particular cell during transcription. From this information, a number of techniques can be performed. So let's take a look at these now. As you might know, analyzing gene expression will typically involve isolating mRNAs from a cell, and from these we can perform reverse transcriptase polymerase chain reaction or rt-pcr. This is where we can take mRNAs and create a complementary DNA strand in essence reverse. Engineering the DNA template would have generated the mRNA during transcription. That is why the enzyme that performs this function is called as reverse transcriptase, and the process is known as reverse of transcription. Whenever we do this, reverse transcriptase will use the mRNAs as a template to generate a single-stranded DNA molecule that is the complement of mRNA. As we recall, mRNAs have a poly A tail or a number of adenine RNA bases in a row. So we can use poly B3 primer or a series of thymine DNA bases to help reverse transcriptase get going. Once completed, another enzyme is used to degrade the mRNAs, and then the DNA polymerase is used to synthesize the complementary DNA strand, resulting in a double-stranded DNA molecule, which we will call as the complementary DNA or cDNA. Now we can examine some of the applications of these approaches. For an example, we want to know precisely when a gene is being expressed during the any growth and developmental stage of an organism. So we could isolate the mRNAs from different stages of the development and perform rt-pcr to get cDNA fragments that corresponds to all of these mRNAs. Then we could amplify a specific cDNA molecule that represents the gene of interest using the polymerase chain reaction by employing a primer that is specific to the gene of interest so that only the targeted cDNA representing the gene of interest is amplified. We can repeat this process for each of the stages of embryonic development we are testing. Here, embryo development is just an example. Then we can perform gel electrophoresis with a column for each stage of the development, whereby only the amplified products will be relevant, since it will be much more abundant than anything else. So if we get a lot of gene of interest, it means that the mRNA is produced was present in the sample during that stage because its presence was necessary in order to get the corresponding cDNA in the first place. So this is how we can tell when a particular gene is being expressed in the embryo. So quite similarly, this approach can be used to determine which tissues within an organism are experiencing a particular gene by extracting mRNAs from different tissues and going through the same process with the cDNA and amplification to see which tissues or sample contain the mRNA associated with the gene of interest. But more importantly, biologists are often interested in understanding genome-wide expression. In other words, we want to understand the ways that Many different genes act together to produce and maintain a functioning organism. To do this, we will typically perform something called as DNA microarray assay. This is a powerful technique involving a huge grid of tiny spots, and in each well sits many copies of a single-stranded DNA fragment called a probe attached to a solid surface which each represents a particular gene. So we can think of this as a grid of many different genes of an organism or even all of them ideally. Then by the process we already discussed, mRNAs that are made in the targeted cells are isolated and reverse transcription is performed to generate the cDNAs. These cDNAs are labeled with fluorescent tags and introduced into the array, allowing them to hybridize with any complementary DNA sequence they can find. Now recall that these cDNAs will have sequences that should be identical to the segments of the genes that produce the mRNAs, because DNA is template for the mRNA during transcription and the mRNA is the template of the cDNA during reverse transcription. So one of the two strands of some cDNA molecule made from an mRNA should bind to the DNA fragment in the particular well that can produce the mRNA in first place. Since they will be highly complementary, and once time is given for hybridization to take place, any cDNAs that exhibits little to no binding is washed away. Whenever binding has occurred, this is indicated by the fluorescence of the cDNA and is clearly visible. If we label different samples with different colors, such as different tissue samples, we can then get an enormous amount of qualitative data all at once. So everywhere you see one color, such as red, red cDNAs are bound, meaning that the particular gene is expressed in the tissues that we label red. Wherever we see green, that the gene is expressed in the tissues that we labeled with green. So if we see yellow color, it means both red and green cDNAs are binding. So we see the intermediate yellow color indicating that the gene is being expressed in both tissues. And if we see little to no coloring, the gene is not expressed in either tissues and no binding will occur. This technique is immensely useful in a variety of contexts. For example, we want to know how gene expression has been altered under water stress conditions. We can compare the mRNAs in a regular cell and water stress cell on the same array, and as simply as identifying a color on a grid, we can know precisely which genes have been altered. Then we can just simply sequence that gene and know precisely where mutation has occurred, and therefore get information about how the resulting protein has been altered, which allows us to understand on a fundamental level why the plant wilting is occurring and suggest new approaches for the treatment. There are a lot more other common applications of these techniques as well, which have a range of utilities. All of this makes the DNA microarray assay an indispensable tool in the molecular biology laboratory.